This is a production of Cornell University.
Super. All right. Thanks very much, Margaret. It's really nice to be invited. I'm very much appreciative. Yes, it's about 5:45 here in the evening. So you guys are just halfway through your day and I'm just trying to get away from my emails and everything. So this is actually a really nice thing to be doing. So as Margaret said, I'm at the University of Southampton. I've been there for about nearly 7 years now. And today I'm going to talk about the domestication work I've been doing. And this is looking primarily at eggplant, but we do have other projects looking at brassicas and some other things. So fingers crossed, let me know if the slides aren't changing or my camera dies or anything. Margaret will do a thumbs up or a thumbs down. So this is my plan for the talk today. I'm going to talk about why domestication is interesting, why I personally get out of bed in the morning and are super excited by this, and also why eggplant, why would we want to study this species? So I'm going to talk about the demography of eggplant origins, which is about where it originated, how many times was the crop domesticated, looking at gene expression divergence. This is using RNA-seq in the last few years. We've done some work looking at selective sweeps in the genome, and we've been zooming in on one of these traits as well, trying to get down to some candidate genes involved in it. Just to quickly go through the three main collaborators that I've been working with. This isn't everyone, but Anna Page, she was a PhD student, graduated about a year ago from my lab. Jane Gibson, who does some bioinformatics. She largely works on human DNA and things like that, but she's done a few clever tricks for me with the bioinformatics. And Rachel, who I was lucky enough to meet just after she published some work on eggplant, and we struck up some ideas about what to follow up with. So why would we study domestication and why would we study eggplants? Well, domestication from is, I think, a really interesting model to study evolution. And it works really well for a few reasons. And these are just some of the reasons I've come up with. So firstly, we pretty much know what, if we don't know what the progenitors are, at least we can know that they will be extant. So we're not looking at speciation over millions of years where various species have been gone extinct at any point. We largely know what traits were involved. So we can zoom in on these rather easily. It tends to be things like shattering, fruit shape, fruit size, taste, colors, things like that. It's recent and it's rapid. So again, we haven't got millions of years of evolution to try and decipher. We've just got a few thousand years of very strong selection. And in a lot of cases, we have parallel evolution. So studying one crop, you can often piggyback off some of the ideas from other crops, potentially. So it's really interesting to study whether parallel evolution has occurred. But at the same time, if crop A has lost the ability to shatter its seeds, maybe crop B has in the same genes. So it's very quick to zoom in on those potential candidate genes. Of course, there is the money side of things. So it's really important to know where our food comes from. And this isn't about sort of farm to fork, that sort of thing, but more about where the species came from, how they originated, which traits were involved and what genes were involved. And when we think about climate change and food security, we're going to have to get more food from less land. So if we can know more about the genes involved in yield and abiotic and biotic tolerances, that's exactly what we need at the moment. So why eggplant? Well, firstly, I think it's just generally a really interesting crop in and of itself. I've got here on the right a picture of a wild eggplant and obviously a domesticated eggplant. The wild eggplant fruits are only a couple of centimeters in diameter, and you can probably see there that there's thorns all over the calyx and the stems as well. So it's gone quite a long way in evolutionary terms to get from the small fruited, very bitter tasting wild eggplant to the very large purple, much more mild tasting eggplants that we would cook ourselves. So I think eggplant is interesting in and of itself, but because it's in the Solanaceae, it's related to several other crops. We've got tomatoes, however you want to do it. I didn't know how I was going to pronounce it. Tomatoes, potatoes, peppers, chilies, and even tobacco as well is in the same family. And for some of the traits and for some of the crops, there's definite parallels in evolution. Fruit size and fruit color and fruit shape are some of the obvious ones, but we've also got changes in plant architecture and things like that. And the tomato research tends to be a little bit further ahead the eggplant, and that's largely because more people eat more tomatoes than they do eggplants, going back to the money side of things. So in eggplant, we can piggyback to a certain degree on some of the work that's been done in tomatoes. And just to highlight this parallel evolution, so I've got here a table from a paper that's almost 20 years old now, doing QTL mapping in eggplants. And there was some significant crossover in the QTLs found in eggplants as there was found in tomatoes. So this is based on the same genomic position. So at the moment, we don't know if the same genes are involved, but there's reasonably good evidence that at least there's some parallels for things like fruit weight, fruit shape, anthocyanins, and a stripe on some of the tomatoes and some of the eggplants as well. So it's a really nice study system for several reasons. So what do we already know? So I started this project in about 2014. And before this, there was a bit of phylogenetic work looking at the origins of eggplant. And this map on the left shows how the different taxa and subspecies, however you want to call it, are related to each other. So in Africa, we have a range of wild eggplants, and it's thought that there was transport to sort of China and India from the north of the range in Africa. And this is where the domestication happens. So that's shown by the yellow star. And this gave rise to the sort of primitive small fruited eggplants, which then went on to be domesticated into the large fruited eggplants. But also from these small fruited eggplants, we have got regression potentially back into being weedy forms. So wild eggplant is often seen as a weed in farmers' fields. And potentially this is actually derived from the primitive domesticates as opposed to being undomesticated true wild eggplant. So Rachel Meyer, as I said before, I've had a lot of contact with her. She did a lot of work looking at domesticated eggplants and hypothesized more than one origin of the domesticates. So she found significant differences between a sort of Western and an Eastern group. So separated into India, Nepal, Pakistan, versus Thailand, Burma, places like that, with a potential third origin in the Philippines or Malaysia in the southeast there. So the first paper that I talked about didn't suggest any more than one origin, but the second paper I've just talked about potentially 2 or even 3 origins. So not only have we got parallel domestication of tomatoes and eggplants, but we might have parallel domestication of different groups of eggplants as well. So why and how have these crops been domesticated? Well, I've already said about the fruit size, and that's one of the sort of dead giveaways that differ. I've got here in the center, I've got a picture of a small part of the plant, and we've got a lot of spines in the top photo. And this is the wild eggplant, of course. It's got these 1 to 2 centimeter long spines on the leaves, the stems, and the calyx, whereas domesticated plants, 99 times out of 100, they don't have the spines at all. And on the right here, I've just got a zoomed in photo to show these spines. And so I always apologize to the undergraduates who are coming to work with me if they end up on an eggplant project, because unless they're solely working on the domesticates, they may well end up with scratches all down the back of their hands when they're harvesting moving plants and things like that. But it's not like every eggplant looks the same. So when you're walking around the supermarkets, you're probably going to see purple eggplants that are about 8 inches long, and they'll all fit through a hole in a machine and everything, and they'll all be very, very similar. But this is some of the diversity that you can find in the landrace eggplants. Apart from the couple of red individuals in the middle, these are all the same species. These are all Solalum melongena, so they're all the same species. So we've got massive differences in sizes, shapes, got colors ranging from almost whitish to green through to yellow and purples and almost reds. Got long, thin eggplants, short, fat, round eggplants, everything in between as well. So it's super diverse compared to what a lot of people might expect from a domesticated species. So what we wanted to know was, using more up-to-date techniques, we wanted to revisit the multiple domestications hypotheses. So was there one or more than one domestication of eggplant? Where did this happen? And to look into a couple of specific aspects of the taxonomy as well. So there's two main things I wanted to investigate. So firstly, we've got this potential for feral eggplants. So are the weedy eggplants that are found in farmers' field actually derived from the domesticates and then they've sort of been rewilded? Or are they just wild eggplants pre-domestication, so to speak? And there's also a group of cultivars which have been either called subspecies ovidurum or Solanum ovidurum on their own. And ovidurum, obviously, is sort of egg-shaped. And they have fruits that are about the size of a chicken's egg, so not very large at all. And some taxonomists have said that this is a separate species. Others have said they're just primitive domesticates. And others have said that they're a specific group of domesticates that humans have selected on since the domestication of eggplant. So we've got a few hypotheses to test. And we wanted to use more sort of genomic scale markers to investigate this, because the research prior to this was either looking at 3 or 4 nuclear genes or chloroplast gene sequences, or they were using AFLPs and a mixture of AFLPs and ITS sequences. So what we did is genotyping by sequencing. So as Margaret can attest, the genome of eggplant is pretty big. It's not huge, but it's not manageable when we were starting up the lab. So we did genotyping by sequencing of 95 accessions. This demography work has been published. I've put a link there as well. And we tried to get a mixture of all the different gene pools. So we've got wild eggplants, we've got Solanum ovidurum, so whether that's primitive or whether that's derived, we wanted to investigate that. And we looked at land races from throughout Indochina. So almost everywhere where the land races are grown in quite large amounts, we looked at those. We stayed away from the sort of highly inbred cultivars, because I think for a lot of cultivars, there's a reasonable chance of them being transported all over the world, strange hybrids being produced that may have different qualities that the breeders are interested. So there's a good chance that any GBS data we got from these plants would just, it wouldn't necessarily be reliable as to the origin of that land race in particular. So we stuck to the land races. We didn't look at the improved varieties. After doing the GBS, we ended up with nearly 5,000 SNPs. We did some phylogenetics and we looked at the population structure as well. So again, trying to address these questions about where and when, or where and how many times the eggplant was domesticated, and these questions about the taxonomy as well that were previously only hypothesized. So this is a phylogenetic tree of the SNP data. And I've broken it down into 3 red boxes, and I'm going to talk about them separately on the next slides. And on the right, we've got structure plots. I'm not going to talk too much about those, but you can see there does seem to be population structure being pulled out. And the legend's at the bottom, but the legend will be on all the slides. So we've got outgroups in black. We've got Solanum insanum, so that's the wild progenitor. That's in light blue. 3 geographic groups of cultivars, Solanum melongena, and purple is the Solanum ovidurum, the egg-shaped fruits. So looking at the wild, so we pulled out a reasonable amount of phylogenetic structure in terms of the wild species. So in blue, we've got Solanum insanum, that's our progenitor species. And then we've got 3 other species that came out in monophyletic groups, Lichtensteinii, Linianum, and Incarnum. And the first 2 are found only in Africa, and the third, Solanum incarnum. And it does get confusing because we've got an incarnum and an insanum, but they are clearly different based on their genetics. But incarnum is found in very North Africa, moving into the Middle East. So this is possibly where the wild progenitor was actually derived from, and that seems to match our phylogenetic work. Something to note, within Insanum, there seems to be two separate groups. I don't know if you see my mouse, but I'm pointing to it now. I've got Margaret nodding, so that's fine. So there's sort of two groups. These two accessions are from India, and then these accessions are a mixture from more like Thailand, Thailand, Vietnam, Philippines, and Malaysia. So it looks like in the wild progenitor, there's two quite different genetic groups, and it looks like the domesticated eggplant is domesticated in the east. So from Malaysia, Thailand, Vietnam, that sort of area. So then we've got this sort of slightly messy group in the phylogeny where, well, it's not even a group really. It's the sort of thing that no one likes to see in a phylogeny because it's very hard to say what's going on here. But we've called this a gradation. So we're moving from very wild-like genetically and phenotypically plants up into the kind of almost looking domestic type plants. And this is where almost all of the ovidurum fall. So all the purple dots, there's about 9 or 10. They pretty much all fall out in this group. So it does suggest that ovidurum is a primitive domesticate. However, we did some approximate Bayesian computation, so testing different phylogenetic hypotheses. And it actually looks like this group, they may be partly because of gene flow. So there may be some hybridization going on. These aren't necessarily just primitive domesticates. They seem to be kind of primitive, but with gene flow from other domesticates. So they're a bit messy in terms of their genomes, basically. But I think we can safely say they're not a derived group, like a single derived group from the domesticates. And there's a couple of other things in here. We've got things that appear to be wild and things that appear to be land races. So there's definitely this gradation between true wild and land race, I think, in eggplants. And because geographically they do overlap, I think it's reasonable to expect that there is some gene flow. And there has been work measuring gene flow, and there are domesticated alleles found in wild populations in some parts of India. So certainly, based on previous hypotheses, I don't think we can say this is unusual. It just seems to be that there's some gene flow messing up any sort of clear genetic groupings here. But then we move into the domesticated groups, and you'll see there's mainly two groups within the domesticates. At the top, we've got almost all the Indian accessions, and at the bottom, we've got Southeast Asia. So the mainland Southeast Asia, as well as the Malay Peninsula, Philippines, and places like that. So it does look like a single origin in or around Malaysia or Vietnam or Thailand. And then these sort of two genetic groups within the cultivated, a more Western group into India and Pakistan, and a more Eastern group where we think the domestication took place. Just two things to highlight. Well, one thing to highlight twice, I guess. In both of these groups, we've got plants that look wild. And these are actually, these four accessions are all ones that were called feral eggplants. So what it does seem to show us is that these feral weedy eggplants that are found in farmers' fields, they do seem to be derived from the domesticates. So I think that kind of answers that question about where they came from and whether they are just true wilds, which I'd say no, they're not, or whether they're derived from the domesticates. And again, we did the ABC, the approximate Bayesian computation. And again, it's the most likely origin of these is through hybridization. So they're potentially domesticates that have escaped cultivation and have ended up hybridizing with wild eggplants and ended up with this sort of hybridy genome. But largely this tells us that the ferals, they're not true wilds. They do seem to be derived from the domesticates, or at least a portion of their genome seem to be domesticated. So just summing that up, so in terms of the demography and where and when all these things happened, it looks like we have a single domestication in Southeast Asia. Solanum ovidurum seems to be a primitive domesticate, but there is some level of hybridization and admixture. And some of the phenotypically wild eggplants do seem to be derived from the domesticates, but they may be derived with some ongoing gene flow as well. So something else we wanted to look at was genes under selection. So as I said before, this is quite often one of the targets of a domestication investigation. So to do this, we did RNA-seq. So when we did the GBS, the genotyping by sequencing, these are random loci from throughout the genome. And I think the chances of more than a few % of those ending up in genes is very slim. So what we wanted to do was actually try and zoom in on genes that may have been under selection. So we sequenced transcriptomes, which are generally a lot smaller than a genome, not necessarily easier to assemble, but at least you know that the reads you get are from coding regions by and large. So for this study, I'm going to talk about 28 individuals. We did sequence some more, but when we sorted out all the phylogenetic work, some of our individuals seemed a bit admixed. So we've avoided those in this story, but we've still got the data. And we looked at the wild eggplant, which is in Sarnham. We only ended up sequencing 4 of those because some of the wilds ended up being hybridy, and we tried to ignore those. We've got 3 of another wild species in Carnum, which we think is the progenitor of Insanum. We looked at 6 of Ovidurum, that's the one with the egg-shaped fruits, and 15 Melongena, the domesticas. We looked only at land races again. And we looked at leaves because if you imagine comparing the transcriptomes of fruit, you've got very large, very small, green, yellow, red fruits and stuff like that. Whereas if you look at the seedlings and you look at the leaves, they're much more similar. So what we're not doing is enriching for things that just happen to be because the fruit is larger, for example. And then you just pull out loads of genes involved in fruit enlargement, which may or may not have been under selection during domestication. So anyway, so this is how it's broken down. So the first thing we did, we measured genetic diversity. And there's about a 50% drop in diversity during eggplant domestication. So, well, 47%. So comparing Insanum to Ovidurum, so this is going from wild to primitive, we've got about 47% reduction in nucleotide diversity. And then we've got another small drop as we get from Ovidurum into the domesticates as well. So this fits all our hypotheses about how domestication works, in that the cultivars or the land races we're looking at today tend to have a subset of the genetic variation present in the wild. And given that we only sampled 4 Insanum and 15 domesticas, this seems a pretty substantial drop as well. We found several fixed differences. So there are potential for these genes to be involved in domestication phenotypes. So about 1,400 during domestication, and then a very small number have fixed differences between Ovidurum and the land races. But if you imagine that there are fixed differences in alleles at a gene involved in pathogen resistance, that absolutely makes sense, that there may be different pathogens in the different environments. Or it may be to do with fruit size, fruit shape, plant stature, things like that. So this amount of drop of diversity is nothing untoward. It actually fits fairly in the middle of other findings. So maize and soybean, the genetic bottleneck is probably less than this, but in rice and common bean, it's probably the bottleneck is probably stronger than that. So eggplant fits in the middle of all of those crops. We did find quite low diversity overall, though. So in the wild progenitor, almost a quarter of the loci were monomorphic, but of course we are zooming in on only coding regions here. So there's no non-coding regions involved. So generally diversity is lower. And we found almost a third of the loci in the land races were monomorphic as well. So that does lead us to a slight problem. So what we wanted to do was look at dropping nucleotide diversity. So compare the nucleotide diversity for every locus where we had enough information, compare the nucleotide diversity in the wild and the nucleotide diversity in the domesticates. And if we see a significant drop, then we can presume that this locus was under selection. The problem is that a third of the loci are monomorphic in the domesticates anyway. So when we actually run tests like this, everything is monomorphic in the domesticates. So actually all we're finding is the genes that have the highest variation in the wild. They don't necessarily correspond to those that have had the greatest drop in diversity because they've all got no diversity at all. So we did follow up on this by doing some genome-wide work, which I'll talk about next. But because this is transcriptomes and we sequenced all the plants on the same day at the same time, they were all grown in the same ways, we could examine new expression divergence too. And I'm going to tell you about two comparisons. So comparing the wild to the land races and comparing the wild to the primitive ovidurum. We've done the other comparisons as well, but I don't really need to go into all the details here. So comparison A, so this is going from wild up to all the domesticates. We found almost, well, over 2,000 loci were differentially expressed. And what was quite interesting was that the majority of these were either down-regulated or silenced. So there wasn't a 50-50 mix of up and down regulation. They were almost all down-regulated. And then when we compared Insanum and Ovidurum, we had almost 1,000 differentially expressed loci. And again, a large number were down-regulated or silenced. So what this makes me think is that if domestication involved gene knockouts, which is what our data seems to suggest for eggplant, this could certainly make things like neo-domestication easier, because if the easiest way to get from a small fruit to a large fruit is to knock out 3 genes, you can pay someone to run CRISPR now and knock out 3 genes. So potentially domestication of other crops or improvement of more primitive crops or less well-studied, less improved crops, it could be relatively easy. I say relatively easy, I mean, it's still CRISPR, it's not going to work first time, etc. But people have done this. So there's some work here by Lemon et al., where they looked at a wild Solanaceae and they knocked out some tomato domestication genes. But they've also tried to re-domesticate tomato as well by knocking out domestication genes. So it's quite interesting, I think, that the majority of genes, at least in eggplant, were down-regulated or silenced. And with CRISPR, you can go in and just knock out genes essentially as and when you want. So I think this is quite interesting with potential for food security and things like that as well. So we looked at all our differentially expressed genes and we measured FST, so a measure of genetic divergence, basically relative genetic divergence between the different populations. And there's 3 comparisons shown here, and I've separated them into differentially expressed, which is DE, of course, and not differentially expressed. And you'll see in all of the comparisons, the differentially expressed genes have a greater relative divergence between the populations than the non-differentially expressed genes. So this tells us that there is a signature of selection at the nucleotide level because we've got an increase in divergence between the 2 populations. So humans have selected for possibly an allele that's differentially expressed, and at the same time that's increased the amount of nucleotide diversity between the populations. So selecting on one allele out of the dozens that might be present would increase your genetic divergence as well. So this follows our sort of hypotheses about how human selection may be working. And of course, we were interested in what these genes were. I've named a few here, but we ran a Go analysis as well, just to see if there was any categories of genes that came out as differentially expressed. And we got some quite interesting Go terms that do fit what we know about domestication. So we've got terms involved in defense. So this is largely this was due to biotic defense, but there was some abiotic tolerance in there as well. We've got jasmonic acid biosynthesis, jasmonic acid being a plant hormone, so it's involved in growth and things like that, and photoperiodism and flowering. And of course, there's a reasonable chance that domestication involves selection on flowering, because farmers will want a field of eggplants that are flowering and fruiting at approximately the same time, rather than what might happen in the wild and a farmer having to go out and pick a few eggplants every day for 3 or 4 months. So these Go terms do make sense. Just zooming in on a couple of genes. This is a couple to do with fungal resistance. And that makes sense because humans would have probably selected on resistant genotypes. And there's also 2 genes here, Gigantea and early flowering 3. And they've been shown in Arabidopsis to be involved in photoperiodism and flowering. And these 2 genes actually directly interact, at least in Arabidopsis. So this is quite a nice little story that we are we're doing a bit more work on this now, trying to get a bit of more information about where and when these genes are being expressed and how that differs between a wild and a domesticated eggplant. I'll just take a breath now. So as I mentioned, when we tried to look at nucleotide diversity on a gene by gene basis, we've got so many monomorphic loci in the eggplant that any tests on a gene by gene basis were simply pulling out high diversity in the wild genes as opposed to candidate domestication genes. But during, or well, towards the end of Anna's PhD, there was a new version of the eggplant genome came out. So the old version was quite highly fragmented, and we didn't try and do genome-guided transcriptomics. But later on, this better, more contiguous genome was released. So what we did is we mapped the RNA-seq reads to the genome, and we used this to look for not individual loci that may have been under selection, but actually chunks of the genome which contain multiple loci with a signature of selection. And we did this by calculating FST, which is what I mentioned on a couple of slides previously, where it's looking at divergence. So high FST between a wild and a domesticated population does suggest adaptive divergence or the influence of human selection. And we measured pi as well, which is nucleotide diversity. And we did it over chunks of the genome of 450 kb, because we think that LD drops off fairly low within about 450 kb, although we don't have great data for that at the moment. So we had to pick a window size, and this fitted and it was functional kind of thing. And just to highlight, so this is not eggplant data, this is just what we're looking for. And so if there's a region of the genome where there's very strong selection, and I've shown it by this red box, we might expect pi to be significantly lower than the rest of the genome because we've lost genetic diversity through a selective sweep. And we might expect FST to be higher than the genome average because we've got more differences between the wild and the domesticated, as expected based on the rest of the genome. But this is an eggplant data, however this is. So this is all the chromosomes stacked up from chromosome 1 to 12 going left to right, alternating between grey and black, grey and black. And I've got FST at the top and pi at the bottom. And pi is actually getting higher as you go further down. FST obviously getting higher as the further up. So what we're looking for is regions of high FST and low pi. And I've highlighted 3 of those regions that came out which had both a high FST and a low pi. They're on 3 different chromosomes, and each is about 4 to 6 megabases long. And if we look at them, we've got the region sizes, sorry, they're between about 6 and 8 megabases long. Each one has several hundred genes in, of course. So because we haven't got transcriptomics from very, very large numbers of plants, we can't really zoom in on very small windows. So we're not saying that there's one gene in each of these 3 loci that we're particularly interested in. We've got over 1,000 genes to follow up, which is nearly 4% of the eggplant genome. So that's quite a lot for us to follow up on. But Anna did do some Go analysis, and there was an overrepresentation of genes involved in photosynthesis, in the biosynthesis of the photosystems, in chlorophyll, and in the chloroplasts. So this is not the same as we found with the RNA-seq differential expression, but it does seem to make sense that humans may have selected on higher productivity, and chances are that involves some regulation of photosynthesis, chlorophyll, chloroplasts, and things like that. But this is just based on 3 regions of the genome and several hundred genes. So we've got a bit more to follow up on before we can say specifically what genes we think are key to the transition from wild to domesticated. But what we have done is zoomed in on one particular trait, which is this spininess that I talked about. So on the left here, I've got a wild and a domesticated eggplant. This isn't the true wild progenitor. This is a species called Lineanum. And at the bottom, we've got the leaves, and they're actually switched round. So the domesticated leaf is on the left with the wild leaf on the right. And the paper I mentioned from 2002, they did a QTL mapping population crossing these two species and generating an F2. And they looked at a number of different traits. They looked at fruit size, fruit shape, fruit color, They looked at aspects of leaf morphology, sizes, shapes, things like that. And they did also look at spininess as well. And as I said before, that's one of the conspicuous differences between wild eggplants and domesticated eggplants. And actually, the eggplant is in a clade called the spiny solanums. So it's not just the wild progenitor that's spiny, but most of the eggplants in this group are spiny eggplants. So anyway, so what we wanted to do was kind of follow up on this and look at it in a bit more detail. This is the QTL map that we were working with on the left. So it's quite a large chunk of the genome. And on the right, this is just slightly more updated. It's looking at the same traits though, and it's looking at the population grown in different environments now. But what you'll notice is that all the different prickles of the phenotypes that were mentioned, a lot of them stack up in this one region of chromosome 6. So we've got spines on the stem, the leaves, the petioles, and the fruit and the flower calyx. They all stack up in one region of the genome. So even though there are smaller QTL all over the genome, there is this one large, one region of the genome that corresponds to a relatively large amount of the genetic variation. And we talked to Marie-Christine Dornay, who is at INRA in France, and she's been selfing this F2 population to generate RILs. So what we did is we got some RILs from her, and we got some original F2s as well, seed from the F2s, and we did some genotyping. And one of the first things we realized was that there's basically a binary switch between presence and absence of spines, and it maps to this region of chromosome 6. And the other QTL all correspond to the size of the spines. So you need to have the wild allele at this QTL from eggplant chromosome 6 to have any spines, and then multiple QTL around the genome will give rise to larger or smaller spines. So what we were doing is we wanted to zoom in on this sort of binary switch almost for the presence absence. So we started out with a 7 megabase region, which is largely based on this genetic mapping. And this is where it's useful having multiple crops, because some of these markers are only found in tomato, but the tomato chromosome 6 and the eggplant chromosome 6, apart from a region at the top, which has been flipped, they're largely collinear. So we could use data from tomato and the data from the eggplant genome, which was coming online while we were doing this. So we figured out this was approximately a 7 megabase window. We generated 120 plants that segregated 3-to-1 spiny to non-spiny. So as I said before, it looks like there's a Mendelian locus controlling presence absence of spines. And then within the spiny plants, there's a gradation from very small couple of millimeter spines up to the sort of 1.5 centimeter spines that scratch the skin on the back of your hand when you get near to them. So what we wanted to do was zoom in on this 7 megabase region. So we generated some transcriptomes from Lineanum. I'm going to talk about that in a second. And we mapped those to the eggplant genome. And then we just simply looked for indel markers. So then I had undergraduates in the lab who were scoring indel markers in these 120 plants. So we had 14 markers in the 7 megabase region, and we've zoomed in on about 2 megabases now. So 2 megabases is a sizable chunk. It's still got a couple of hundred genes in it, we think, but we have narrowed it down a bit. We're only looking at 120 plants. So I don't think, based on those 120, I don't think it's worth us throwing any more markers at it, because we haven't got enough recombination in that area of the genome. But what we did do is we collected some plants that were, well, we collected the fruit from some heterozygous plants. And we've got, from one of those, we've got about 1,000 seed. And from another couple of plants, we've got several hundred seeds. So what we can do now is grow up the large populations. As Margaret will attest, an eggplant is a pretty sizable plant. So growing up 1,000 plants is no mean feat. However, you can tell whether the plants are spiny or not spiny when they're only about 2 or 3 months old and only 6 inches tall. So this is something we need obviously time, and we need someone to be able to do this. but it is something that we're going to start next year, I think. So as I said, we generated linearnum transcriptomes. And what we did is we did 6 transcriptomes. We did 3 from the spines and 3 from the veins, because the spines on the leaves are formed out of the veins. So what we wanted to do was look at differential expression between essentially a spine, which is an elongated bit of a vein, and another piece of vein. And as I said, we did it for three different linearnum plants. We did differential expression to look at the genes that might be involved in this trait. This is just the sort of typical plot you see, but clearly we've got separation of the 3 veins on the right, and no, sorry, the 3 veins on the left and the 3 spines on the right. And these are all our differentially expressed genes. And so they fall out in 2 main clusters. We've got high expression in the veins, low expression in the spines, or vice versa. So at the moment, we can't really say whether it might be a knockout or an upregulation or anything like that. We found about 0.6% of transcripts for DE, so quite a small number, which is easier to work with. And about 50 of these fell out in this 2 megabase region. So I think what we secretly wanted was 1 or 2 genes in that 2 megabase region to also be differentially expressed. Sadly, we got about 50. But some of them we can follow up on, and there's a few interesting candidates. There's genes involved in the modification of cellulose, and of course, you've got cell walls being elongated, so that makes sense. And other genes that may or may not be involved. And then there's a bunch of other genes as well. So we're not ready to say we've got 1 or even 5 or even 20 candidates, but we have got enough data that we've got things to zoom in on. And I think once we grow up our larger mapping population, hopefully we can zoom in on a smaller region of the genome. And then we've got a bit more to sort of, we can bring together our data a bit better to compare the differential expression. We can look at where they map, and ideally, finding potentially differentially expressed genes in this region. Although we don't know it's differentially expressed, it might just be a polymorphism that changes the coding sequence. We've got no a priori reason to think it's differential expression, but again, it's a different place to look. And just recently, there was a Solanum linearnum genome released. It's not the most high quality, but we've mapped it back to the domesticated eggplant genome. And so we've now very easily, we can go in and design hundreds of molecular markers just for this small region. We just need time and we need a bit of space to get on with this right now. But we're quite happy, we're quite enjoying this at the moment. I've had various people working on it, and we seem to be getting closer anyway. So just to sum up, I think I'm about 40 minutes, so that's not bad. So we've got an interesting and a convoluted domestication of eggplant. We've got post-domestication gene flow, and we do have a lot of overlap between where these land races are grown and where the wild eggplants are found. So it's not unexpected. We've got escape from domestication. So we've got quite an interesting potential evolutionary story about de-domestication and whether you get how this kind of reversal of the domestication syndrome comes about. Lots of candidate genes. So we've got a lot to work with. It's a fun time to be working on eggplants, but at the same time, it's this sort of giant Excel sheets and R documents with hundreds of different things in that we need to look at. But we do seem to be getting closer, at least to this one trait that we're particularly interested in. So that's quite exciting. And we are sort of zooming in on it right now. So just to acknowledge people, so Anna was the PhD student who did a lot of the demography. Jane helped us out with some of the more tricky bioinformatics. And Rachel, she supplied us with some seed. I think she may have illegally sent it to us through the post, but we grew it and we threw the plants away. We didn't grow it in our own gardens. I've had a few undergraduates and master's students working on this, too many to name, but a lot of those undergraduates have been trawling through the Indel PCR and stuff. Special thanks to Marie-Christine Dornay at INRA, because she didn't just supply the seed, but she has been maintaining these populations. And she's obviously supplied a lot to Margaret as well. So she's still involved and she's still interested, which is great. I had funding from the university when I got there. I got an annual adventures grant. I'd like to thank Margaret as well for the invite. It's really, really interesting to talk to you guys. And thanks Cornell as well for hosting as well. I'd obviously be happy to take any questions. I presume that's where we're going next. Is that right?
Yes. Thank you, Mark, for a wonderful seminar. And thank you to the Zoom gods for a smooth connection.
Amazing, like 5,000 miles away and no Zoom problems.
Right. Great. So I'll open it up to questions. You can either type it in the chat or you can just unmute yourself and ask your question. I see there is a question here. Developmentally, how do the spines form? And is the absence of spines due to a failure of specification? Cool.
I don't think we know. I mean, I think once we get closer to knowing what the candidate gene might be, or what the sort of presence absence gene might be, I think that's going to help. They do seem to just grow out of the veins. It's hard to say exactly what tissues or exactly what cell types are involved. I think we need to do a bit of work on that. I have been talking to people about doing electron microscopy of the developing spines. That was a conversation that started about a year ago, and obviously we all know what happened about 9 months ago. So that's put on hold for now. I think we would like to know a bit more, and I think it will help us find candidate genes as well. So if we know it's an elongation of the cell wall, or if we know it's a breakdown in some compound that would normally constrain the cell wall, we can start zooming in on that, whether it's lignin or something like that. But developmentally, they don't seem to be the same as trichomes in tomatoes, which is a bit disappointing because obviously if it was the same developmentally, we've got candidate genes we can start looking at. But I don't think we can do that because they don't seem to be developmentally the same as a trichome.
They're multicellular, right?
Yeah, they're multicellular, yeah.
Cool. Yeah, Adrian, the professor who asked the question, works on live cell imaging. So that would be a great system to do live imaging on.
Okay, yeah, so, sounds good, yeah.
A question from Jeff Doyle. Are you working on more of the messy gene flow issues using network methods, perhaps?
Yeah, well, we have actually been doing that. So we've got, at the moment, we've got a project on brassicas, and the postdoc who's working on that, she's now getting very adept at all these network type, doing gene-based phylogenies and putting them together to make networks and things like that. So yeah, we are following up with that actually. This was published over a year ago, and since then we've got a lot of the brassica data, and turns out brassica's a real mess as well. So actually the postdoc's probably going to apply some of their skills to this eggplant data.
Very cool. I have a question, and then Hamid has a question. I'll ask him to unmute himself for his because it's a two-part, if he's okay with that. But I'll start. I'm interested in those feral kind of weedy species that you identified. Do you think that they're like F1 hybrids or like really close to the hybridization event and thus exhibiting hybrid vigor, and that kind of helps them be successful as weedy species? Or is it more of a situation where they've kind of hybridized several generations ago and then gone through?
Yeah, I don't know. I don't think these are sort of early generation hybrids that are super heterotic and then just manage to live everywhere. We did a bit of work which we couldn't really conclude much from, so I'm hesitant to talk about it too much. So take this all with a pinch of salt. But what we did is we looked in these feral, we had some transcriptomes from the ferals, and it seems like a non-insignificant proportion of the loci that are introgressed do seem to be candidates for being under selection during domestication. So they have a largely domesticated genome, and the bit that's introgressed seems to be enriched for genes that may have been under selection during domestication. So it may be spiny genes, dwarfing genes, small fruit genes that have introgressed into the domesticates. but we've got a long way to go with that. So pinch of salt.
Yeah. Very cool. Hamid, are you okay asking your
Yes.
question? Thank you.
Sure. Great talk, Mark. Thank you.
Thanks, Hamid.
I have two questions. One is, you mentioned that the Ovigerome has gene flow with the cultivated plants. So I wonder if you have tested for gene flow with the wild ancestor also.
Yeah, let me try and think about that. I think it's entirely probable that has happened. I guess that, as far as I remember, that wasn't one of our scenarios we tested, though.
Sure.
Yeah, I don't think we've tested that, so I couldn't really comment on that. But I think there is a chance. Yeah, absolutely.
And I was also wondering whether the fruit phenotypes are diverse in Ovigerome, or do they all have egg-shaped?
No, so everything that's called Ovigerome is based on having these egg-shaped fruits. So phenotypically, they are all very similar. There's some sort of yellowish fruits and some very bright white fruits, but because there's so much diversity, there's long and short and fat and round and everything. I think everything has just been named Ovigerome that looks about like that.
Sure.
In our phylogeny, even though they're all in that gradation, I think there's two Ovigerome accessions that are in the domesticates. So it looks like they may be independently derived egg-shaped fruit cultivars. But I think they're found in very different places in the phylogeny and very different from where the other Ovigeromes are.
Sure. Great, thank you.
You're welcome.
And we have one more question from Jim Giovanoni. Given the absence of spines, Jim's wondering if you might expect something other than a cell wall candidate, for example, a transcription factor or kinase or some other kind of master regulator. And perhaps you could take your mapping region and compare it to a non-spiny relative, like tomato, to narrow.
Yeah, actually, so I've got a PhD student starting after Christmas who is going to be zooming in on this. So we're going to use as much data as we can get. So we've got one of the Lineanum genomes. We've got all the transcriptomes that we've generated. We've got a small number of domesticated genomes, and then we've got things like tomatoes and even potatoes if we want to go that far. So what we'd like to do is try and throw together as much data as possible. It's interesting you mentioned kinases, because in that region of the genome, there are a few kinases. I know that kinase is a very broad topic or a broad category of genes, but there does seem to be a cluster of kinases. So that might be entirely circumspect and nothing to do with spines, but it does seem that there is a cluster of kinases in there.
I'm excited to find out the spinniness.
Yeah, give me a year or two. I'm not quite ready, but yeah.
Yeah, great. So if there are no more questions, join me in thanking Mark for a wonderful talk, and TGI Friday, Mark, you're done with your day. So thanks.
Well, no, I've got to talk to some PhD students now, apparently, in about 10 minutes. Is that right?
Oh, great. Yes. Okay. PhD students, join Mark. You're not off the hook yet.
It's fine. No problem.
Great. Thank you so much.
Thank you. I've really enjoyed it.
This has been a production of Cornell University, on the web at cornell.edu.